We've been talking a lot about traits that are dominant and recessive. Dominant traits being ones that are always expressed, recessive traits being traits that need 2 copies of that allele in order to be expressed. But in reality, for most traits, for most phenotypes, they're not usually simple dominant recessive and only one gene controlling that phenotype. Things are a little bit more complicated. So let's go into some of those kind of variations or extensions of this dominant and recessive inheritance pattern that are more commonly seen in humans and lots of other organisms to determine phenotype. So this first idea is the idea of what we call incomplete dominance, where neither allele is completely dominant over the phenotype. And what I mean by that is that each allele has it's this like blending effect. Each allele will contribute a portion to that final phenotype, and that results in kind of a halfway point between the two alleles, between the two phenotypes, right? So let's say this is actually pretty common in flowers, flower color. Red, we have 2 copies of allele. We have a red allele and we have a white allele. So CR being the red allele and CW being the white allele. So if an individual has 2 copies of that red allele, they will both produce red pigment and that flower will be red. If an individual has 2 copies of the white allele, they will both produce a lack of pigment, or white color, I guess you could say, a white pigment, and that flower will be white. But what if we get a heterozygous individual? What if we have an individual that has both a red allele and a white allele? Well, the red is not dominant to the white, and the white is not dominant to the red. They are incompletely dominant. So 50% of the color that will be produced in this flower will be red, and 50% of the pigment will be white. And what do we get when we mix red and white? We get pink, right? So each allele is going to contribute an equal percentage to the phenotype, but they blend to those 2 alleles blend together to create a middle, or a middle phenotype, you could say, between the 2 extremes of either allele. This is slightly different, very subtly different than what we call codominance. So codominance, instead of blending, we have distinct effects, individual impacts of each allele that an individual has. So in humans, a great example of this is blood type. So in codominance, we have multiple A and B alleles and what we call the O allele or the I allele. But when we have 2 alleles of different types combined together, that doesn't result in a third distinct phenotype, right? So let's break this down. So we have 3 alleles for blood type. We have IA, IB, and I. So these represent, what these represent is the type of protein that will be expressed on the surface of a red blood cell. One of many different types of proteins on the red blood cell. We just track this one, okay? So if an individual has 2 copies of this A allele, this IA allele, they will have type A blood type. Or if they have the IA allele and then just this I allele, they will still have type A blood type, because this I allele right here essentially codes for a lack of a protein. No protein is expressed essentially at this point. Okay? So it's kind of recessive in the sense that it's a lack of a protein is what it codes for. So if you have 1 copy of this A allele, you'll produce that A type protein. And if your second allele is this I allele, it doesn't matter. You're still going to have type A blood, because this allele right here just says you're already expressing this type A protein. Okay, so each allele has a distinct effect on the final phenotype. So same thing for type B blood. You can be homozygous for this B allele, or you can be heterozygous, where one of your alleles is this IB blood type and the other allele is this I, this lack of a protein, right? But what if you get type A allele and type B allele? This is how we get type AB blood. So each allele is expressed equally and has a distinct effect on the final phenotype, the protein on that surface of that red blood cell. So you will get both type A proteins and type B proteins. Okay, so this is different than incomplete dominance, because in incomplete dominance this would result in kind of a mix between those two proteins, but instead we get both proteins are expressed. And then type O blood as well, right? So if somebody has type O blood, this protein does not get expressed. There is no protein expressed at this location on red blood cells. And the only way to get that is to have two of these I alleles, these lack of a protein alleles, basically. So here we have an example of having multiple alleles. So we've only really been looking at traits that have two alleles, right? But here we have three alleles that govern phenotype, and they have a slightly different inheritance pattern of this codominance, where they each have a distinct effect or distinct impact on that final phenotype. So let's practice with this. Would it be possible for two parents who have type B blood, so their phenotype is type B blood, to have a child that has type O blood? Okay, so both parents have type B blood. Could they have a child with type O blood? Well, let's see. What genotypes did we just look at could result in type B blood phenotype? Right, so these parents could be heterozygous, have one B allele and that other I, that lack of a protein allele, or they could be homozygous and have two of those type B proteins, two of those type B alleles. Let's do some test crosses. Do we think any of these combinations, so this is parent 1, parent 2, would any of these combinations, do you think, result in a child that has type O blood? Remember, to have type O blood, you gotta have those two I alleles, not IB, not IA, just those I alleles. Okay. Do we see any of these crosses that could potentially result in two of those I alleles coming together? If you can't see this, that's totally fine. Just do these 3 test crosses real quick. It'll take you like a minute. So let's do one. Okay, so if we have one parent that's heterozygous and the other is homozygous, do we get any type O blood? No, they would all have type B blood. Okay, so that doesn't work. What if they're both homozygous? So both parents are homozygous with a type B allele. They are all type B blood. Okay, so that doesn't work either. All right, and then our last combination, what if both of our parents were type B blood, but they're heterozygous, right? They had the type B allele, but then they also had the I allele, meaning that they don't express a protein, that this allele does not code for a protein at that location. So if they both have this heterozygous combination, this heterozygous genotype, do we see 50% of their gametes would have that type B allele, 50% of the gametes would have the type I allele, or the O-type blood, basically. So we have 25% chance of B, so 75% of their offspring here would have type B blood. But look at this one right here, right? There's a 25% chance that if both parents are heterozygous for their blood type, that they could have a child that has type O blood. So again, we've been talking about mostly up to this point dominant recessive, and we've been talking usually about traits that are controlled by one gene, okay? But in reality, for a lot of traits, we have many genes that go into resulting in that final phenotype. So many genes contribute to that final phenotype, okay? Like skin color, hair color, eye color, height. A lot of traits of humans are what we call polygenic traits or polygenic inheritance, meaning that we have many genes at different loci across our chromosomes that all contribute to our final phenotype. So let's go through an example. So let's use skin color, a simplified example of skin color, how skin color is inherited, as an example of polygenic inheritance and how we can combine these having multiple alleles and incomplete dominance. So skin color, how do we get the pigmentation in our skin, right? We have these cells in our skin called melanocytes that produce pigment molecules, pigment proteins, basically, right? And in humans, one genotype is not necessarily dominant over another genotype. So these melanocytes can produce many different types of pigments, okay? One melanocyte can produce both what we call eumelanin and pheomelanin. So pheomelanin is more of like yellow and red pigmentation. Eumelanin is more like black or brown pigmentation. Okay, so this melanocyte can produce both colors, and it's kind of like that flower color. They can blend together. Okay, so we see this idea of incomplete dominance in human skin color. We also have multiple alleles for skin color. The melanin that we produce, again, we have eumelanin, which is typically brown or black in color, and pheomelanin, which are red and yellow in color. So we have at least 4 different, it's actually much more than this. This is kind of a simplification of this process, but we have at least 4 different alleles that all contribute to producing our final skin color. And on top of that, not only do we have many different types of alleles that produce different variations of skin color, humans actually have over 300 genes, 300 loci, that the combination of all of these genes with all of their own unique set of alleles, sometimes more than 2 alleles at each loci, that all combine to produce skin color. So, in polygenic inheritance, when there are multiple genes contributing to a final phenotype, we tend to see a continuum of phenotypes. Before, we'd been talking about a kind of a binary, like yes or no, short legs, long legs, has ocular albinism, does not have ocular albinism. When we talk about a continuum, it's a spectrum from very, very, very dark to hardly any pigmentation at all, okay? And so this is because each loci, each of these over 300 different loci and all of their own alleles combine incomplete dominance and codominance into producing that final phenotype. Okay, so it's usually a little bit more complicated than a simple dominant recessive. A lot of traits, especially in humans, are polygenic, which is why we have a wide spectrum of phenotypes. In addition to that, obviously our genes are interacting with the environment, and genes can be turned on or turned off or alter their expression, what we call being a plastic gene. Plastic meaning it can have multiple phenotypes depending on the environment that it's in. So that makes things even more complicated, right? So let's talk about this for a minute and talk about how genes are influenced by their environment. We have genes that are influenced by our environment, right? Like skin color. We're exposed to sunlight, and typically our melanocytes are going to produce more pigmentation. So that's an example of how our phenotype can be plastic. It can change depending on the environment. A lot of organisms have genes that change their expression depending on the environment, depending on environmental cues, right? So like, let's look at the snowshoe hare right here. Right? What is wrong with the snowshoe hare? What is happening? So the snowshoe hare in the wintertime will have a white coat because it's going to be a snowy environment, and white blends in pretty well with that snowy environment, right? But as soon as the snow starts to melt, it warms up, gets moving into springtime, that white coat will stand out against this really dark environment, right? So it actually switches its coat color depending on the time of year. And so snowshoe hares have over 600 genes that all control their final coat color, okay? But some of these genes change their expression in response to day length, in response to how much sunlight how many hours of daylight this snowshoe hare is getting, right? And it's this really cool, complicated process where these circadian rhythm proteins typically degrade and are produced on like this 24-hour clock. But exposure to sunlight can change that process, can alter how quickly or slowly these proteins degrade or are produced. So same set of genes, 632 genes, but they're going to produce different products based on the time of year. So they're reacting to changes in their environment. Okay, so there's, again, just drive this home, there's no such thing as nature versus nurture. It's nature and nurture, right? It's genes and environment. It's not either or.